Welcome, everyone. Thank you for joining us for today's keynote presentation titled Emerging Drug Combinations, Evolution from Preclinical Discovery to Clinical Development. It's my pleasure to introduce to you Dr. Khyati Shah. Dr. Shah received her Ph.D. in Molecular Pharmacology from the University of the Pacific in Stockton, California. Currently, she's a postdoctoral research scholar at the University of California, San Francisco. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. My name is Antonina Pavlenko, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your question into the drop-down box. Our speaker will respond to your questions via email following the presentation. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Khyati Shah. I will now turn the presentation over to her.
Hello, everyone. My name is Khyati Shah, and I'm today going to give a keynote presentation on emerging the drug combination, how it evolves from preclinical discovery to clinical development. So for this purpose, I would like to give an outline of my talk. First, I will be talking about why precision medicine is so important in drug discovery. However, it's been a long time that we have been practicing the precision medicine to discover the better drugs. But there are some inadequacies that exist, and I would highlight what are those and how we see that the limited therapeutic responses or innate resistance is the main bottleneck of being successful in truly practicing the precision medicine. So the way we take our approach is we design the rational combination therapy first to deepen the therapeutic response and use that as a method to overcome the therapeutic resistance. So in the preclinical laboratory setting, we think that this is a very successful method, and we have shown this over again and again. So I will go over the examples of that. And at the end, I will even discuss how it can be integrated into the clinical trials and what is the current landscape and future vision for the combination therapy in the clinic. All right, so let's just get started. Traditionally, the drug discovery process is made to have one size fit all. We have a patient population that has very different genotype or different phenotype. And mainly the way earlier clinical trials, especially the original trials with chemotherapy in cancers were designed, that most of the patient gets all the drug disregarding their genetic or phenotypic underlying cause. So what that cause is, that was an adverse event in some patient. Some patient population had no benefit of the drug and some patient had benefit. So the precision medicine is all about how we can identify the patient population that derives the benefit of the therapy. And the way we do this is to practice a stratified medicine by identifying a genomic or a molecular marker present in these patient population and stratifying the patient population with that and practicing integrating a companion diagnostic biomarker with it to identify which population can get benefit from these therapies. And the classic example of this is in case of a breast cancer population, 65% patient population have a molecular marker called estrogen receptor. So these patients likely to derive benefit from the drugs that are designed to inhibit the estrogen receptor. 25% of the patient population displayed HER2 positivity, and these patients will derive a benefit from HER2 targeted therapies. And some although we understand the genomic etiology or genetic makeup of these, we don't have a classical molecular marker linked to this population, and those are called triple negative population. So overall, this is a very well-known practice in precision medicine to identify the patient population based on the molecular marker. And hence, the precision medicine ensures the delivery of right intervention to the right patient at a right time. So to take this further, we designed a mechanism-based therapy. So I will talk about estrogen receptors in this case. Estrogen receptor, when binds to estrogen, leads to a dimerization. This recruits the co-activator at the targeted estrogen receptor regulatory element, and it leads to increase in the proliferation and ultimately the breast cancer growth. So this is a genetic driver. Estrogen receptor is a genetic driver in the case of breast cancer in some patients. That's why selective estrogen receptor modulars were designed so that it binds to the estrogen receptor and leads to recruitment of co-repressor, and this further suppresses the gene transcription, downstream gene transcription, and hence leads to inhibition of cell growth in the case of breast cancer. So that is why we practice the mechanism-based cancer therapy. So this leads to a question that then we should be really successful for the patient whom we identify that are having a positive molecular marker such as estrogen receptors. So is that the case? If you look at over here, most of the drugs that were identified for that particular molecular target or a given indication listed here, we see that the patient have modest benefit, meaning that their progression-free survival is not longer and their overall objective response in the patient is also limited. Almost no patient over here received like 100% cure indicated by the violet bars over here. This leads to a question that why we see this reduction in the objective response rate, and what clinical factors informs us about this. So the major way to identify this is to look at the clinical trials that are conducted for these therapies. And almost this is a generic form of a waterfall curve of the patient whose tumors are measured after giving a standard therapy for a given timeline. What you can see that some of the patient on the left side indicated in the red and black part, they do not respond, meaning that they go on to have a progressive disease. Some patients, however, do respond, but they almost never have a complete response. Very, very few patients in a clinical trial display complete response, meaning that their tumors are completely eradicated. So the main limitation on why we are not successful in practicing the precision medicine is because A, the problem with innate resistance, that despite having a molecular target, despite knowing their genetic makeup, and despite giving the right drug, that we don't understand why these patients have innate resistance. And although they do respond, they never receive a complete response. They have a residual disease or incomplete drug response. So these two are the main two bottleneck of being successful in our drug discovery process. And in case of a tamoxifen, we can use this as a standard example and try to identify what are changes that are happening in these tamoxifen sensitive cells. So most of the estrogen receptor positive cells, when they receive tamoxifen, they undergo cell death and most of the tumor shrink. And that can be indicated over here in the cell culture. You can see that in presence of MCF7, which is the estrogen receptor positive cells, if you give them no treatment, these cells grow. In presence of estrogen, they grow even faster. And tamoxifen, which is the estrogen receptor blocker, it prevents the transcription of classical estrogen regulated genes, and you see that tamoxifen is effective in inducing cell death in these, or reducing cell proliferation in these MCF7 positive cells. However, there are tamoxifen innate resistant cells. These cells are known to have a growth factor signaling, which is centered around AKT and PI3 kinase pathway. When these pathways are activated in these estrogen receptor positive cells, these patients do not derive any benefit from tamoxifen treatment, meaning that they display innate resistance. So we have the cell line models to test this, and the model for that is GR751. When you give them no treatment, they grow as similar as in presence of estrogen, as well as in terms of tamoxifen, meaning that now, despite having estrogen receptors, the tamoxifen has no efficacy on these cells, maybe because they have a higher AKT activity. So we wanted to study this further, and these are the published results in molecular cancer research. And what we identified, we looked at different AKT isoform, and we found that AKT1 and AKT3 are significantly associated with the patient who have a recurrent tumor. And when the AKT1 and AKT3 isoforms were also correlated with higher aggressiveness, these patients had more chances that there would be a relapse upon therapy, and they will have even more aggressive tumors. So clearly AKT signaling plays an important role in this. To study this in detail, what we did is we designed a particular cell line models to study this in detail. And when we take our MCF7 cells, which are classical estrogen receptor positive cells, what I showed that they respond beautifully to tamoxifen, when we overexpress AKT1 and AKT3 exogenously, we see that there is a downstream activation indicated by higher phospho-GSK 3-beta activity. And these cell lines, when engrafted in xenograft, we see that in presence of tamoxifen, they grow even faster in MCF7 cells, despite they being estrogen receptor positive cells. So we can use this and identify what is the contribution of AKT in governing a tamoxifen response. We took this study further to identify how different AKT isoforms plays an important role. So I have already shown you in presence of in MCF7 cells, tamoxifen reduces the cell proliferation. When we have this exogenously AKT overexpressing cells, you can see that tamoxifen induces growth in case of the AKT1, as well as in presence of AKT3. This is very similar behavior to estrogen receptor negative cell lines such as MDAMB468, which is known to have higher AKT1 and 3 isoform, and MDAMB231, which is known to have a higher AKT3. And I have already shown you the tamoxifen insensitive cell lines that also has estrogen receptor but has no efficacy in terms of with the tamoxifen responses. So clearly this suggests that AKT signaling plays an important role, and specifically AKT1 and AKT3 isoforms are main contributors in limiting the therapeutic benefit of the tamoxifen. So this leads us to ask a big question in the field. Can we identify differentially rewired signaling network that alters tamoxifen responses? Can we also design a synergistic combination therapy to deepen the tamoxifen responses? And will there be any clinical benefit to tamoxifen-resistant patient, especially identifying a surrogate marker predicts the resistance and identifying a secondary drug target that helps us to overcome the resistance? So to do this, we performed a gene expression analysis. We specially used the microarray technology in AKT3 overexpressing cells, which were treated with tamoxifen, and compared with the MCF7 cells, which are treatment sensitive. And what we found is the top deregulated genes indicated over here in red, and top downregulated genes indicated in green in the TAKT3 MCF7 panels. When we studied this further and validated in our qPCR assay, we found that the fold changes were very similar, and we were able to validate what we see in our gene expression analysis. Moreover, when we take this gene signature, which were upregulated in AKT3 tamoxifen treated cells, we found that having higher expression predicts a lower survival of the patients in the patient database using Oncomine and KM plot data set. Also, the downregulated genes, when their expression is high, that predicts that the patient have better survival rate, meaning that our signature has significance in terms of governing how these patients respond on tamoxifen treatment. And when we look at the cellular network or cellular pathways that these genes drive, we found that cell proliferation as well as the cell motility function was governed by a very interesting gene called RRM2, and we took this further in our study. So I will introduce what is ribonucleotide reductase. Every cells have the ribonucleotide phosphate that needs to be converted into deoxy ribonucleotide phosphate, and this reaction is catalyzed by an enzyme called ribonucleotide reductase. This enzyme activity is required for DNA synthesis, and ribonucleotide reductase itself has two units, the large unit RRM1 indicated here in green and a smaller unit RRM2 indicated in orange. This complex itself functions as a catalytic enzymes for providing the DNA synthesis. And hence, this is a very important step in any cellular growth or in any cancer proliferation. RRM2 activity increases at the late G1 and early S phase interface because that's where the synthesis starts. Also, if you look at the literature evidence, there are various evidence indicating the importance of this RRM2 enzyme in governing the genomic integrity and DNA repair. It's been shown that the SCF cyclin F ubiquitin ligase governs the RRM2 degradation, and it is responsible for creating a genomic stability. However, when there is a genotoxic stress or there is a DNA damage that occurs, RRM2 expression is stabilized, and hence the DNTP production is increased and the cell is likely to repair their DNA. So this shows that RRM2 complex has an important role in governing the genomic integrity. And in case of a chemotherapy-induced DNA damage, this plays an important role because if the cells are able to efficiently repair their DNA, they are most likely to avoid the cell death in case of a cancer, and hence RRM2 could be one of the potential target. It's been also shown that suppression of nucleotide metabolism by removing the RRM2 is also responsible for oncogene-induced senescence. It's been shown that RRM2 via the E2F-mediated transcription factor is responsible for inducing the senescence in when you have overexpression of oncogenes such as RAS. But if you superphysiologically express the RRM2, these cells are able to undergo the transformation and cause cancer. So it can overcome the oncogene-induced senescence. And hence, RRM2 plays a central role in governing the response to most of the chemotherapy drugs. But how that is related to estrogen receptor, and we wanted to establish this correlation. So we examined the different database submitted in the Oncomine to look at the expression of RRM2 between estrogen receptor positive and estrogen receptor negative cells. And what we found that in estrogen receptor negative tumors, RRM2 expression was highly expressed. So as in our cell line models, MCF7 being our sensitive cell line model, which has higher amount of estrogen receptor alpha indicated over here by higher ER alpha 66, had low RRM2 level. But other cell line models which exhibit diminished response to tamoxifen, such as AKT1, AKT3, MDAMB468, ZR751, and MDAMB231, they known to have a higher expression of RRM2, and they have diminished expression of classical estrogen receptor alpha 66. indicating that there would be some correlation between estrogen receptor expression and RRM2 expression. To expand this further, we took a panel of estrogen receptor positive cell lines, which are indicated over here by presence of estrogen receptor alpha 66. Only the ZR751, which I showed you earlier, is tamoxifen insensitive cell line, shown to have higher RRM2 expression. And in case of estrogen receptor negative cell lines, in this panel of 6 different cell lines, we found that all of them had higher RRM2 and they were estrogen receptor negative mainly. So clearly indicating that RRM2 and estrogen receptor negativity correlates, and whenever you have higher RRM2, probably these cell lines have diminished expression of estrogen receptor alpha and hence won't get any benefit from tamoxifen. So can we use this RRM2 as a target to enhance the response to tamoxifen? And to do that, what we did is we performed the siRNA knockdown in our panel of cell lines, MCF7 being tamoxifen sensitive and RRM2 low, and the other cell line panels were RRM2 high. And we used 2 different siRNA, and what's shown over here in AKT1 and AKT3, overexpressing cell lines, that we were able to successfully knock down the expression of RRM2. And what we can see is that MCF7 cells knocking down and treating with tamoxifen in MCF7 cells diminished the cell proliferation, but not in other AKT cells. When you treat them with the RRM2 siRNA, there was no effect on cell proliferation. However, you note here that when you knock down the RRM2 and treat them with tamoxifen, they were able to induce, there was a reduction in cell proliferation with the RRM2 knockdown and tamoxifen treatment. So if you compare the red and the green bar, there is significant reduction when you remove the RRM2 from the equation. So this is a very interesting observation, but as we know that certain siRNA-based studies has to be validated using a pharmacological inhibitor, and hence we use the Didox, which is a ribonucleotide reductase inhibitor, whose molecular structure is given over here. This was done in collaboration with a company called Molecules for Health, invented by Dr. Alfred Howard, who synthesizes ribonucleotide reductase inhibitor, and is mainly Didox has multifaceted action. It's mainly used as antiviral. It has antibacterial property. It mainly works by anti-inflammatory, and it also reduces the NF-kappa B mediated signaling. This drug is mainly used in terms of for sickle cell anemia and in case of a certain T-cell malignancies, but its role in breast cancer was not known. There are clinical trials that was done initially to identify the activity of the drug. Patients were able to tolerate 6 grams of drug, but this drug as a single agent had no efficacy. It is a tyrosyl radical scavenger, and hence it serves as an anti-inflammatory, and it destabilizes the RRM2 subunit of ribonucleotide reductase enzyme. So we use this in our in vitro as well as our in vivo model of tamoxifen resistance. In case of MCF7 cells, just the control treatment, we see there is a cell proliferation. Tamoxifen was sensitive only in MCF7 cells, but not in these AKT high cells. Didox as a single agent had no efficacy, but the combination did a much better job in reducing the cell proliferation in this in vitro model. Similarly, when we take our AKT3 model and made the xenograft out of it, we see that the tamoxifen induced even more pronounced tumor growth of these tumors, and Didox as a single agent had no efficacy, but the combination did much better job in reducing the tumor volume in these AKT3 overexpressing MCF7 cells, which is our in vivo model for tamoxifen resistance. So clearly this suggests that Didox do have significant benefits in patients. And we wanted to even understand how overexpression of RRM2 confers resistance. So to do that, we took our MCF7, which is estrogen receptor positive and tamoxifen sensitive cell line, overexpressed them with the two different constructs of RRM2. And what we see over here is that there is a colony from the colony formation assay that in the MCF7 cells, tamoxifen is able to reduce the cell proliferation shown in the panel 1. But when we overexpress the RRM2, there is no reduction in the tamoxifen. Tamoxifen loses its effect. And when you combine them with the tamoxifen and Didox at a given dose, we see that this is able to reduce the cell proliferation significantly. And this is also indicated, the reason why we see this synergy is because the ribonucleotide reductase enzyme activity of these cells when we overexpress RRM2 is significantly higher, but Didox reduces this ribonucleotide reductase activity much efficiently, and these cells now don't have any DNA to proliferate, and hence we see this synergistic effect, clearly indicating that how RRM2 plays an important role in cell proliferation in case of a cancer. To study this further, we took our RRM2 overexpressing cell lines and tried to identify by performing cancer 10 signaling pathway reporter array to identify which pathways that RRM2 drives. And what we found is when we overexpress RRM2, there is a significant increase in NF-kappa B and HIF1 alpha activity. And when we treat these RRM2 overexpressing cells with Didox, we see the complete reduction in this NF-kappa B activity, indicating that by inhibiting RRM2, we are inhibiting multiple different signaling pathway. It also restored the P53 signaling. And when we look at the effect on cell motility, just overexpressing RRM2 increases the cell motility and decreases the percentage open area. But when we treat them with Didox, this was able to inhibit the cell motility and cell migration as well as cell invasion. So next, we wanted to identify if RRM2 plays an important role in the acquired resistance. When you have the estrogen receptors, positive cells, most of the cells respond to tamoxifen. But what happens in the clinic when the patient undergo long-term tamoxifen treatment therapy is they have certain mutations, amplification, or epigenetic changes that are occurring in their tumor, and that renders them ultimately irresponsive to tamoxifen. And we wanted to identify if RRM2 has any important role in this setting as well. So to determine the clinical relevance of RRM2 expression, what we did is we again went back to Oncomine and identify a patient database which were treated with tamoxifen and were indicated as either tamoxifen sensitive or tamoxifen resistant. And in these patients, what we identified is that RRM2 mRNA or expression level were significantly higher in the patient who were tamoxifen resistant. And if you look at the recurrence-free survival in these estrogen receptor positive patients, having high RRM2 predicted the lower survival and regression-free survival as well as lower overall survival, meaning that RRM2 plays an important role in how patients respond to the therapy. So to do this further, to study this further, we know that RRM2 limits the benefits of tamoxifen. We made the acquired resistant models by taking MCF7 cells, which is the tamoxifen sensitive cell line, treated them with the appropriate doses of tamoxifen in a stepwise increasing manner to come to our drug-tolerant persister cells, and then expanded the drug-tolerant persister cells to resistant cell lines. And we made 4 different acquired cell lines from 4 different estrogen receptor sensitive cell lines. And we observed that these cell lines, when they become acquired resistant, they downregulate the expression of classical estrogen receptor alpha 66, and they overexpress RRM2. So you can see over here that RRM2 is highly expressed in tamoxifen resistant cells, indicated by TEM-R over here. So at that time, the interesting study that came out, which indicated that there is not only classical estrogen receptor that is present, there is also a splice variants of estrogen receptors, such as estrogen receptor alpha 46 and estrogen receptor alpha 36. Estrogen receptor alpha 36 was mainly interesting because it's a splice variant. It is made by alternative splicing, where the exon 7 and 8 were not transcribed, but the 9 is transcribed, and it had a lower molecular weight. So estrogen receptor alpha 66 is a classical 66 kilodalton molecular weight protein, whereas estrogen alpha 36 is 36 molecular weight protein. The role of estrogen receptor alpha 36 was also determined that it dimerizes at the plasma membrane with the estrogen, with the growth factor signaling, and in presence of tamoxifen, these are known to drive MEP kinase and AKT pathway and ultimately leads to cell proliferation. So we looked at our estrogen receptor positive and estrogen receptor negative cell lines. So these estrogen receptor positive cell lines were mainly positive for estrogen receptor alpha, and only ZR751, which is the tamoxifen insensitive cell line, had higher expression of RRM2. Interestingly, these cells also had lower expression of estrogen receptor alpha 36. But the estrogen receptor negative cell line had significant higher expression of estrogen receptor alpha 36 and higher expression of RRM2, indicating that there is an inverse relation between RRM2 expression with classical estrogen receptor alpha, but there is a direct relation with the estrogen receptor alpha 36 with RRM2. And probably because of this, when tamoxifen is present, it causes the cell proliferation, and patients do not derive any benefit from because of having higher estrogen alpha 36. So to study this further, we wanted to see the changes, molecular changes that happen over a period of time. And hence we took the MCF7 cell lines and just allowed them to grow or treat them with the 5 micromole of tamoxifen. And what you can see is having a higher confluency cells and treating them with the high dose of tamoxifen, most of the cells die by the day 10. However, by day 15, they are able to start their cell cycle and they start growing again. And when we look at the molecular changes that happens over here, by the day 15, they are able to grow and have higher RRM2, meaning that they are probably able to start their DNA synthesis and ultimately the cell cycle. If we look at the corresponding changes in estrogen receptor signaling, classical estrogen receptor alpha 66 goes down, whereas the estrogen alpha 36, which is responsible for diminishing response of tamoxifen, starts to come up. And these cells also have corresponding increase in the growth factor signaling, such as EGFR, HER2, and higher phospho-ERG, as well as higher phospho-AKT activity in them. If we look at canonical NF-kappa B signaling, there is also increase in P65 phosphorylation and RAL-B component as compared to C-RAL. So this indicates that as the cells become acquired resistant, they also upregulate NF-kappa B activity. As well as these cells have higher expression of BCL2, which is a classical NF-kappa, which is a classical anti-apoptotic gene and diminished response of PARP as well as caspase 9 signaling. So can we use this prior knowledge that we acquired that RRM2 is helpful in sensitizing the innate resistant cells, which has higher AKT, and use that to design a strategy for abrogating acquired resistance? And to do that, we look at the molecular events that are happening in MCF7 cells, in tamoxifen-resistant cells, and tamoxifen-resistant cells when we treat them with Didox. So what we found is when we treat them with the Didox, which is the ribonucleotide reductase inhibitor, we see that there's a downregulation of RRM2. Correspondingly, the tamoxifen-resistant cells displayed decreased estrogen receptor alpha 66 and increased estrogen receptor alpha 36, which is now reversed upon Didox treatment in combination with tamoxifen. There was reduction in EGFR as well as HER2 signaling when we treat them with Didox. There was reduction in NF-kappa B signaling as well as REL-B expression. And there was a restoration of induction of cleave part and caspase 9, indicating that when you treat them with Didox, when you inhibit this RRM2 activity, these cells are now able to retain the classical tamoxifen response and undergo cell death because there is a restoration of estrogen receptor alpha 66, which is a classical drug target for tamoxifen. So next, we examined the effect of the monotherapy on MCF7 cells, which are estrogen receptor positive and are treated with tamoxifen as a single agent. What you can see over here is that at the day 10, most of the cells die, and ultimately these cells are able to survive the higher concentration of tamoxifen and were able to grow. But when you treat these cells with the Didox, you see that these cells are not able to grow by the end of day 15, and they don't grow at all by day 30. And when you treat the tamoxifen-resistant cells, which are already growing in presence of tamoxifen, and co-treat them with Didox, you see that these cells also undergo massive cell death and cell proliferation. This was also found. We found a similar result in G47D cell line, which is also estrogen receptor and tamoxifen sensitive cell lines, that the combination of Didox prevents the emergence of resistance, but as well as it prevents the growth of already acquired resistant cells. So I would like to summarize my results. What I've shown you is that when the tumors are tamoxifen dependent, they have higher expression of estrogen receptor alpha 66, and they have low RRM2. When these cells become acquired resistant, they start to upregulate their RRM2 expression. These cells show diminished classical estrogen receptor alpha 66 expression and increased in estrogen receptor alpha 36 expression. These cells also have higher growth factor and NF-kappa B signaling, suggesting that RRM2 is a very good target and should be given in a combination therapy to restore the sensitivity of the drug. And this leads to a tumor regression and increased tumor-free survival in the patient. So what I have described until now over here is a preclinical tool of how and why we should use the combination therapy. This forms a very good rationale to prevent the adaptive resistance as well as restore the sensitivity and deepen the responses in the patient. But there are, as we know that if we look at a bigger picture, there are various hallmarks of cancer, not only the genomic driver, but these tumors also have classical hallmark of cancer indicated over here and illustrated by Han and Weinberg. And there are various inhibitors that are designed to act on each of these individual hallmarks. But however, what this suggests is that there's one tumor and there are a combination of these events that are going on. So just giving a single driver is not, or single drug therapy is not sufficient, and the cells will often adapt to rely on the other mechanisms, and hence we should actively use the combination therapy to prevent this adaptation from happening. And in our lab, we think these approaches in terms of how we can use the experimental approaches to target the drug resistance, and we design mainly genomics tools. So what today I told you how we can use the transcriptional changes or microarray or RNA profiling data to create to come to a combination therapy. We can also use the proteomic basis approaches such as reverse RPPA assay or MIB or MS-based mass spectrometry-based studies to identify what proteomics level changes are happening. And also we can use a pharmacological tool such as drug screen to identify different alternative and adaptive drug targets. And ultimately this leads to a design of a combination therapy that we can use to overcome the adaptive resistance that is happening. And in my research, I particularly use these genomic tools or proteomics tools or pharmacological tools listed over here, and we have published in the journal. So you feel free to go to the publication and use them for your advantage or cite them if you find it useful. But coming back to current approaches, so the main limitation why we see this translation is limited from preclinical discovery to clinical transition is because if you look at the current landscape of clinical trials, there are 2,669 clinical trials going on for the targeted therapy. And for immunotherapy, we have about 1,218 clinical trials going on. Of these, 1,494 are the combination therapy trial. So this is the data of the clinical trials that are registered between 2014 and 2015. And if you see, majority of the clinical trials are sponsored by academia centers or cancer centers, about 57%. Industry has very limited, but again, the major role, and there is only 3% combination clinical trials are sponsored by NIH. And of those clinical trials, if we look, 85% are between the combination of immunotherapy and very limited between targeted therapy and immunotherapy. Industry are sponsoring most of the trial, but if you look at the blue data at the end, most of the clinical trial are sponsored by a single company. You might have two targets that for which you don't have a drugs in your pipeline. So you need to foster a collaboration. But such two company collaborations for doing the combination trial therapy is very limited. There are many regulatory challenges and logistic challenges to come up and to have versatile clinical trials for this combination therapy. And hence we need a better system where we can have the leading edge science, where we can have the access to the patient data, as well as we identify and integrate very well the preclinical knowledge that is generated from academic center as well as from industry to come up with a rational combination therapy and identify a potential biomarker and enroll the patient based on who are likely to benefit from the combination therapy. And hence the vision is that this will highly impact and lead to a potential company partnership to have two drugs work in tandem and have a longer therapeutic benefit for the patient. So this calls for what I call is aggregating actionable knowledge, where scientists, clinical researcher, physician, and patient lead to a development of a rapid learning system where we can use our prior knowledge of molecular disease and create an efficient combination therapy to generate better trials, design a better treatment, generate a good guidelines, and ultimately lead to advantage in the patient for curing the cancer. So with that, I would like to say thank you and give acknowledgement to all my team members at UCSF and at the University of the Pacific with whom I work in collaboration. And I thank you all for your attention, and I will be happy to take any questions. Here is my email address. Feel free to email me if you have any questions. Thank you.
Thank you, Dr. Shah, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. As a final reminder, Dr. Shah will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we'll see you next time. Goodbye.